Hello, I'm Cihan Taştan from Üsküdar University Transgenic Cell Technologies Application and Research Center. And today what I'm going to talk about is actually related with CRISPR gene editing, especially for the nuclease editing with base editor or the prime editor site. When we look at the how CRISPR gene editing toolbox improving, especially in 2017 and 2019, there are two different nuclease editor technologies combined with CRISPR-Cas system. One is the base editor, especially in that cases, the nuclease-Cas9 enzyme is joined with the cytosine deaminase or the adenosine deaminase to change the cytosine or the adenine to the thymine or the guanine. On the other side, in 2019, what we have seen that especially prime editor system is improved with the nuclease-Cas9 enzyme. At that case, is joined with the reverse transcriptase. At that case, especially, we instead of changing the single nucleotide, we could change or insert any deleted nucleotide or combination of them. When we have a look at the detail of this two technology, we see that cytosine deaminase and adenosine deaminase systems are working together, and we see that for the cytosine deaminase, the CRISPR-Cas system especially use the nuclease system. And with the help of nuclease-Cas9, instead of producing the double-strand break on the DNA, because it can produce the off-target mutations, at that case, that just produce the nick breakage on the single strand of the DNA. And with the help of the cytosine deaminase, the cytosine can be converted into the urine. And in the later cases, especially in DNA replication, this urine will be the thymine. On the other hand, when we look at the adenosine deaminase, in that case, especially the DNA replication help to inosine to change into the guanine. And with the help of this two base editor system, especially for the single nucleotide polymorphisms, single nucleotide mutations causing the rare diseases can be fixed with the help of these single base editor systems. And when we look at the detail, especially there are kind of the complications. And one complication is that not only the targeted nucleotide, but also the bystander nucleotides can be also converted. And as you can see here that for the cytosine deaminase, you can see that the adenine can be converted, yes, but however, the bystander adenine can be also converted into the guanine. And the same cases is also true for the cytosine deaminase, and you can see that the targeted cytosine can be converted to the thymine. However, the bystander cytosine can be also converted to thymine, and that kind of the limitations are still getting trouble in the base editor system. However, especially for the targeted nucleotide, with the help of without any DNA double-strand break, especially this helps to fix any mutations. So how we can design the guide RNA for the CRISPR-Cas base editor system, especially benchling.com is the free user platform. You can enter any DNA or raw DNA sequences into that system, or you can find the gene of the interest from the system, and this system help us to find what kind of guide RNAs can be useful for you. For example, for here, I entered one of the example. I entered the Duchenne muscular dystrophy gene, DMD gene, and I selected the genome as a Homo sapiens genome. And then for the PAM sequences, which is the protospacer adjacent motif, which is important for the recognition of Cas9, and for the Cas9 PAM sequences, I selected NGG sequences. And after that cases, what we have seen that if you wanted to change or repair any mutations, single nucleotide mutations on the exon side, so you can select the exonic sequences, and the system automatically detect all the base editor potential guide RNAs. On the right side, you can see that the red boxes, and that red boxes are the new amino acids after the base editor system can change the nucleotide, which might be either cytosine deaminase or the adenosine deaminase. And this is up to you, especially if you wanted to change any variants or the mutation takes place in a genome. This could be the old vision how you can change the nucleotides to new amino acids. This is not only the human cells, but also the plant and other animal cells could be the useful applications, because if you wanted to produce the targeted mutagenesis, this kind of technology might be useful to increase the mutations or the capacity of the physiology or function of the protein. And you can see here that not only one nucleotide change, but also you can combine this cytosine deaminase and the adenosine deaminase at the same time on the one base editor system. At that case, what happened, not only change the cytosine to thymine, you can at the same time change the adenine to the guanine. And this kind of combinations can increase your mutagenesis, or you can increase your repairing capacity. On the other hand, with the help of this cytosine deaminase system, you can even delete certain strand of the DNA from just single strand, because the base editor system produced the nick breakage. And on the other hand, what I'm going to come with is the prime editing system. And at that cases, all the game is changing. And what you can see here, that with the cytosine deaminase and adenosine deaminase, the critical point is the DNA replication, because with the help of this base editor system, you can either change the cytosine to urine or the adenosine to inosine. And this inosine and urine is not usual in the DNA sequences or for the genome. And what we have should see that the DNA replication of the cell. And after replication takes place in the cell, then this uridine can convert it into the thymine, or this inosine can be converted into the guanine. And that cycle should be completed with this replication of the cell. And that time you can either change the cytosine to thymine or the guanine, adenine to the guanine, or you can combine these two enzyme capability in a single base editor enzyme if you wanted to change both of them at the same time. And the next question will come that not only to change the single nucleotide polymorphism to edit any mutagenesis, what about to insert the sequences or delete single sequences from that target? And this kind of applications were not applicable for base editor. That's why in 2019, especially prime editing system is developed. And with the help of this system, you can do anything similar to the base editor system. And you can even improve the insertion and deletion of the sequences with the reverse transcriptase domain. And you can see here that DNA changes are not limited for the genome. And instead of using nuclease-Cas9, you can change this Cas9 system because Cas9 can bind to the DNA sequences. You can change Cas9 to Cas13. And this Cas13, as we know that they can bind to the RNA. And by combining the Cas13 with the cytosine deaminase or adenosine deaminase, you can change this nucleotides on the RNA sequences, and this can increase the exon inclusion or the other variable splicing actions. And that's why not only the DNA repairing, but also RNA modifications can be applicable with the help of base editor systems. And on the other hand, using the prime editor system, you are now limited to DNA. However, with the new improvements, this Cas13 might be joined with the reverse transcriptase. At that time, you cannot change the DNA sequences. However, it might help to increase the RNA efficiency based on the results. And what we have come for the base editors, especially these systems getting improving because there are many different limitations. So when we look at the CRISPR-Cas system, especially not only the nuclease limitations, but also delivery, detection, and preparations are the other limitations. However, when we focus on the base editor disadvantages, there are kind of the obstacles. One of the obstacles is that the base editing product purity. Purity means that because there are the bystander editing and the other unwanted nucleotide modifications, the purity of this targeted nucleotide change is not stable or not improved yet. However, on the other hand, the generation of insertion deletion mutations or off-target editing are still getting improved because this nuclease-Cas9 systems, although it only produced a nick breakage on the single-strand DNA, still there is some possibility to produce the off-target editing. And that's why when we look at the limitations of the base editor system, there are still some way to going on. And next slide, let's look at the base editor system to compare with the HDR system. Although there are some limitations on base editor, when we compare with homology-directed repair system, especially base editor has still some advantage. One of the advantage is that when you need to change any nucleotide on the DNA with the help of HDR system, you need to also add the donor DNA into the cell. However, for the base editor system, you do not need to have any more the DNA sequences as a reference sequence. That's why with the only base editor system, the single guide RNA and the base editor nucleases are enough to edit any targeted sequences. And that's why still base editor system has a great advantage and great mutation or modification capacity rather than the classic Cas9 HDR systems. Let's look at the prime editor system, and at that cases, what we are going to use instead of cytosine deaminase and adenosine deaminase, now we combine reverse transcriptase within nuclease-Cas9. And at that cases, our guide RNA sequences are also getting changed. And in that case, while we are producing the 20 to 25 spacer sequences. This spacer sequences will be joined with the guide RNA scaffold as the conventional sequences, and that cases, this sequence are also joined with two other sequences. One is primer binding site, the other one is the RTT, reverse transcriptase template, and these two sequences are also in this RNA form, and that produce the guide RNA as prime editing guide RNA, namely pegRNA. And that pegRNA will help reverse transcriptase to change, to convert the PBS and RTT sequences into the DNA platform, and then the recombination takes place between these converted DNA sequences into the genome. And why this system is getting very popular, because with respect to the homology directed repair, in that cases we were using the donor DNA to change the targeted sequences. Here we do not need any donor DNA, because we insert the donor DNA sequences into the pegRNA sequences as a primer binding site and reverse transcriptase template. And when you design these PBS and RTT sequences in the pegRNA, you are enough to start your gene modification. And you can see here that similar to base editor, the nickase system produced a single nickage on the single strand of DNA, and the other strand can be combined or can be recombined with the PBS and RTT sequences. And the studies have shown that up to 89% of all the variants, all the mutations detected in the human genome can be fixed with the help of prime editor system. And you can see here that not only the transition mutations, but also transversion mutations can be fixed with the help of prime editing. And not only like the base editor, but also the prime editor system could insert or delete any targeted sequences, up to 40 base pair, and you can combine each of these modifications on your pegRNA sequences in the PBS and RTT site. And with the help of that system, not only change the single nucleotide polymorphism, you can even insert any or delete any targeted sequences in a single pegRNA. That's why this system getting improved and getting popular, especially for the rare diseases. We know that there is 75,000 of different pathological mutations taking place in human genome, and this caused the 7,000 rare diseases. And 89% of these genetic abnormalities can be fixed based on the prime editing system, because transitions and transversion of the nucleotides are able to be fixed with this system. And also 40 base pair of the deletions or insertions can be takes place with the help of this prime editor system. So how we can design the pegRNA, especially there are kind of different websites or databases. One of them is the primeedit.nygenome.org, and in that cases you have two potential capacity. One is you can produce the pegRNA to make the disease models in the animals. On the other hand, if you wanted to focus to repair any mutations in the human genome, then you can produce or you can select the pegRNA on that databases with the help of therapeutic gene editing. And in both cases, the database produced the pegRNA with the help of spacer sequence to the specific site of the variation, and then PBS and RTT sequences at the same time to change to your targeted variations. And how you can go on that website, you can see that after enter the website, you can see Prime Editing Designer, and you can select this click, and then you will show that the gene symbol or the clinical variant ID. And especially for clinical variant databases, you can find any mutations causing the genetic abnormality. And after you select the numbers of these clinical variants, you can add the gene symbol or the clinical variant ID into the system. And then you can change your aim, either therapeutic or modeling. And if you wanted to repair the mutations, you can select the therapeutic action, and then you can change the off-target score. Especially Prime Editing system is similar to base editor system, has the almost 300 times less off-target mutation capacity rather than the conventional Cas9 system. That's why these systems are getting popular to be entered into clinical stages. And in that example, I selected DMD gene, and when I entered to the DMD gene, the database has produced more than 700 different pegRNAs specific to different pathological variants previously determined, and the all pegRNAs previously designed to be studied. And you can see here that there are kind of different details for each pegRNAs, and you can see that the reference nucleotides was thymine. However, you can change this timing from the cytosine, and this pegRNA database produced the sgRNA sequences and then PBS and RTT sequences with the help of the conversion of cytosine to the thymine. And that's why you can either select the right side of this website. This is a very big website, especially it produced all the data for your pegRNA before you're starting experiments. And you can see here that the efficiency, the binding capability, and the off-target mutations can be also determined based on the algorithm in the databases. And we used especially this system. We designed several guide pegRNAs on that system. In my laboratory, especially, we started in the first time that we focus on spinal muscular atrophy. And in the SMA disease, especially, there are two different genes. One is the survival motor neuron gene 1, the other one is SMN2 gene. And what we have done that by changing SMN2 gene into the SMN1 gene with the help of pegRNA system. To do that, especially, we designed a couple of pegRNAs and encode this in the lentiviral system. And then we transduce the cells to change the single nucleotide polymorphism to mimic the SMN1 gene, because in the SMA patients especially, SMN1 gene is mutated and a loss of functions takes place, and only the SMN protein produced by the SMN2 gene, however, the production is not enough. And by doing the prime editing system in the first time for the SMA disease, we could show that this SMN2 gene changed into the SMN1 gene and permanently started to produce SMN protein. And you can see here that this is the flow of cytometry charts, and you can see that the GFP encoding cells, which is the biomarker for the prime editor system, especially, you can find this kind of prime editing and base editing plasmid DNAs in the gene databases, and this is also got from the gene website. And then we cloned the PEG RNAs into that vectors, and after transduction of the cells, we follow up to 14 to 21 days, and we could show that the SMN protein, after the changing single nucleotide sequences in the SMN2 gene, we could increase the protein expression from 1.7% up to the 91%. And that's why the prime editing system now getting very interesting, because this system is also permanent gene modifications, and with the help of improved and less off-target capacity, and without any unwanted modifications, this is the future for the clinical trials. And you can see here that the gene knockout and the gene replacement with the homology-directed repair are getting a change into the two different gene repair system. One is the base editing, the other one is the prime editing system. Both systems are applicable for the genetic modifications, especially for single nucleotide polymorphisms, not only for the human disease, but also plant and animal mutagenesis studies are getting important, because you can see here that when we look at the plant traits or the crop traits, you can use the base addition systems to make the nucleotide exchange, and with the help of this system, you can improve the herbicide resistance. On the other hand, you can also produce the targeted mutagenesis in the modified hormone genes in the tomato, or the chromosome eliminations in the maize. And that's why, with the help of maize base editing and the prime editing systems, you can do any of the targeted mutagenesis, not only in the patients but also in the plants. And when we look at the system, how we can develop the better platforms on the base editor or the prime editor system, you can change the Cas9 sequences, or you can codon optimize the Cas9 sequences. Instead of using Cas9, you can use the CPF1 or the other Cas9 orthologs. On the other hand, we can combine the cytosine deaminase or adenosine deaminase with the other deaminase systems, and you can also change the reverse transcriptase on the prime editor enzyme from different virus genome. And the delivery is very important, not only for base editor but also for the prime editor systems. And prime editing system especially has a very long DNA sequences. That's why, for example, adeno-associated virus is not enough to carry any prime editing system. Maybe dual adeno-associated virus delivery systems could be useful for prime editing. However, for base editor system, which is much useful, however, Cas9 sequences are still very long. Instead of using Cas9, maybe you can change these base editor systems or nuclease editing systems with CPF1 like proteins, because they have the very short amino acid sequences. On the other hand, we also need to improve the analysis, because after using the base editing or nuclease editing system, the genotyping or phenotyping of the cells or tissue are very important, and we need to also determine any unwanted editing after this two nuclease editing system. So after we come up with all improvement and optimization, we are going to be ready for clinical qualification of base editor or prime editor systems for the 7,000 different rare diseases. And thank you very much for listening to me. I hope these two kind of the toolbox are be useful for your studies.